This is actually my second effort at this muscle proteins presentation, and the reason for that is I was not sharing my screen, but I made it several slides in, but not sharing my screen. Okay, so muscle proteins can be a little bit confusing because of the terminology and the jargon that's utilized. I'm going to toggle back and forth between not this slide, but the next two slides. What's important to take away from muscle fibers is that muscles are composed of bundles of single large cells. Muscle fibers are formed by cell fusion and contain multiple nuclei. And your major parts of these myofibrils are bundles of the proteins actin and myosin. These are two filamentous proteins. These are the fibrous rather than globular proteins. Now, this picture right here is pretty awesome because of the fact that what you're looking at there is you're looking at proteins. This is probably taken with a light microscope, and you can see the different regions that are being described here. I like this image because this model that's shown below, you can see the definition of these different things. For instance, the Z disc, which is a barrier that we'll talk about in just a little while. Lo and behold, this right here is the Z disc. This right here is a Z disc. An I band, well, it spans from this point to this point. An A band spans from here to here. Now, these are composed of the proteins actin and myosin. The I band contains only actin. A bands contain actin and myosin. The myosin and actin filaments overlap in peripheral regions of the A bands. The H zone contains only myosin. The actin filaments are attached at their plus ends to the Z disc, which includes the cross-linking protein alpha-actinin, and the myosin filaments are anchored at the M line, which is the M disc, which is in the direct center here, in the middle of a sarcomere. Okay, so all of that jargon and all that terminology is worthwhile and helpful to define the players. Now, this next picture, I think, is a very helpful rendering of that because it's kind of taking this image right here and then extracting a little bit of information from it. So I'm going to jump back and forth between this slide and the previous slide. Now, molecules of the protein titin extend from the Z disc to the M line and act as springs to keep myosin filaments centered in the sarcomere. That would be this protein right here. So we have titin, which extends from the Z disc, where I put an X, to the M line, where I put an arrow. We've also got titin going from here to here. Now, the thing is, titin is the largest known protein. Titin is, we looked at it previously, I think it's 37,000 amino acids. This is an extremely long protein, and it acts as a spring to basically keep those myosin filaments anchored in one particular location. Molecules of another protein known as nebulin extend from the Z disc and are thought to determine the length of the associated actin filaments. Okay, so nebulin is this blue protein that's kind of wrapped around this corn cob looking protein known as actin. Now, with that known, let's go ahead and take another snapshot, another look at one of these sarcomeres, because this entire thing from Z disc to Z disc is a sarcomere. So the way that I like to think about this is I like to think about what are the proteins anchored at these different positions. I think, and what is helpful for me is to say actin is anchored at each one of my Z discs. So I have actin, which is tightly associated with or bound to my Z disc. My M line, my middle line of my sarcomere, from there, I have myosin. And these proteins basically overlap one another. Now, what we'll investigate a little bit further in the future is the interaction between myosin and actin. As you can see, these myosin head groups come in contact with actin here, 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 and here. But then they also, which you'll see here, is they also are not connected to that, or there is not contact between actin and myosin. So now to do what I said I was going to do, toggle back and forth between this slide and the previous slide, what we have is Z-disc and Z-disc. So I'm going to scribble out the part of this complicated picture that I kind of don't really want to focus on right now. You can see it very faintly. But there are proteins here and there are proteins here. And those proteins are actin. Now, there's this dense region right here, which I'm going to erase some of my arrows. This darker region right here, that's actually where actin and myosin overlap. So I'll draw actin only, actin and myosin, and then this region in what is known as the H zone is only myosin. So to use the same terminology, the I, A, and H, or if you're a United airline flyer, you're familiar with the IH as Bush Intercontinental Airport. So this region right here, then this region right here, this region is my I band. Now let's look back at this previous slide. The A bands also contain thick myosin filaments. So this would be kind of our, and actually let me jump back for a second. So the A band spans the entire A band from one end of myosin to the opposite end of myosin. So this entire thing is my A band, that entire region. This small cutout here, oopsies, this small cutout here, let me erase that part too and draw a more narrow line. That is my H zone. So I, A, H. So the I has only actin. And I'm writing these things out because these are going to be relevant to what's on your exam and pertinent to specific questions. A is actin and myosin, but also just myosin. But the H zone is kind of the region of it's a specific region of the A band that is just myosin. So I band, A band, and H zone. What are the proteins that are present there? So a question that I could ask is, in what region of a sarcomere, or in which band or zone, do actin and myosin filaments interact with one another? And the correct answer to that would be your A band. Now, in which region does myosin not interact with anything? The correct answer for that would be your H zone. Okay. Then it's also important to remember which protein is anchored to which region or which line or disc. Now, your major components of your myofibrils are your thick actin filaments, as well as your actin and your myosin proteins. Those are the two proteins that are very important. Actin is a protein that participates in many important cellular processes, including mobility of a cell, muscle contraction, cell division, vesicle and organelle movement, cell signaling, cell shape, and many of the processes that are mediated by extensive and intimate interactions of actin with cellular membranes. So this is a protein that does a lot of things, does a lot of kind of, I would describe as gross things, major movements, actin's going to be involved, major changes for a cell, actin's going to be involved. Now, G-actin is a bilobal globular protein that binds adenosine as a nucleotide. Now, what this is able to do ultimately is actin is a series of globular proteins that aggregate together to make that long filamentous structure. So what's a little bit kind of confusing is when you look at this right here, when you look at actin, these little circles here that I think about as looking like little corn kernels, those are globular proteins. Those globular proteins kind of stack on top of one another and ultimately lead to forming these polymers called F-actin and make something that, well, first looks like this and then looks like this. Now, F-actin ultimately resembles a double helix. These polymers have a polarity. That means they have a positive and a negative end. And what they are is they are ultimately attached at the plus ends to the Z discs. So actin, like I said, is involved in lots of kind of gross movements. There's one thing known as actin treadmilling, in which case actin is able to move along or move things along within a cell. A variety of actin proteins can bind, or a variety of proteins can bind actin and basically be migrated from one part of the cell to another part of the cell, kind of treating actin, I don't know if I would say as a treadmill, but, you know, maybe more of a road of sorts. So actin filaments can undergo a process known as treadmilling, where the filament length remains relatively constant, while the actin monomers add at the plus end and dissociate from the negative end. Cross-linking proteins organize actin filaments into bundles or networks, and the actin protein alpha-actinin binds actin filaments into parallel bundles, which hold bundles far apart to allow interaction with myosin. Now, it's this interaction with myosin that we're really going to get after. So what I want you to know so far is I want you to know the confines of one sarcomere, Z disc to Z disc, or Z disc to M line to Z disc. What are the proteins that are associated with that? And now here's where we're getting to kind of the interesting stuff. So the tail is a helical coiled coil with a 7-residue non-polar pseudo-repeat. A and D are non-polar amino acids. So we already talked about this structure when we were talking about filamentous proteins, how the positioning of those A and D amino acids are in close proximity to one another so that they can kind of stabilize this structure. Now, with respect to the structure of this, we have what's a heavy chain and a light chain of these proteins, and those heavy chain and light chains are probably similar to what we saw when we talked about alpha keratin. So we had the same kind of rotation where we have these non-polar residues interacting with one another. Now, a single myosin head structure, this head structure right here, this heavy chain, is what we're interested in. That's where the N-termini of this polypeptide is found. This head structure has a couple of different things. One thing that it has is an ATP binding site. Another thing is that it has an actin binding region. So we're talking about myosin being able to bind to actin. So we've got an actin binding region, we've got an ATP binding site, and then the rest of it is this neck domain with these two light chains. And you know, why on earth do we have an ATP binding site? Peculiar. So the actin and myosin complex which is going to be present in the A band. We're going to have many, many small movements create a very gross large movement. So what we're seeing is, I think, best represented. I mean, this is a great sequence of pictures. So here's our Z disc, M line, Z disc. And slowly but surely, the Z disc moves inward. And then again, moves inward, again, inward. And so these two sarcomeres shrink quite a bit. Now, what's happening there is we're having a transition from contraction of a muscle to relaxation of a muscle. And what we're observing is these two Z discs getting closer to one another. During muscle contraction, each sarcomere is going to shorten, bringing those Z discs closer together. Visually, there's no change in the width of the A band, but both the I bands and the H zone almost completely disappear. So remember, the A band is the region of our overlap between, or basically the A band is from the end of our two myosin kind of filaments. Well, what's happening is that peripheral region where our two proteins are interacting with one another, sure, that's actually kind of growing. The overlapping region is, but the A band as a whole is not. This is caused by actin and myosin filaments sliding past one another. Thus, the actin filaments move into the A band and the H zone. So here's the sequence of events that are taking place. On the right-hand side, we've got that basically the steps kind of detailed. We have nucleotide binding, hydrolysis, inorganic phosphate release, and ADP release. So ATP binding is going to cause the release of our myosin head group from actin. So what we've got in this first picture is our myosin head group that is bound to this green actin subunit. As soon as this myosin head group binds ATP, it releases from this green subunit. So as soon as that release takes place, now actin and myosin at that particular location are not interacting. What's going to happen next is that ATP is going to be hydrolyzed into ADP and PI, or inorganic phosphate. That head group is going to move and find a new actin subunit to bind to. So it's going to adjust, it's going to release the inorganic phosphate as it's bound to this new actin filament. ADP is going to be released and after this movement. And so now what's able to happen is, again, basically return to the top and start it all over again. So myosin binds ATP, releases from actin, hydrolyzes ATP, finds a new binding site, releases inorganic phosphate, releases ADP, and it starts all over again. Now, what this is doing is, you can kind of think about it like here, let's say my fist is the head group. It's going to release, move forward, bind a new site, and it's going to, the myosin head group is going to stay in the same location. But let's use my left arm as myosin head group binds, ATP releases. My myosin head group is going to remain stationary. It's going to bind, sorry, it's bound ATP, so it's released. It's going to adjust and bind in a new location. It's going to hydrolyze that ATP, and what it's going to then do is cause a shift of my left arm, which is my actin filament. It will hydrolyze the ATP, releasing the inorganic phosphate, release the ADP, and again, it's able to bind ATP, release. So we have this series of events where myosin remains stationary. Her myosin does not move, but instead it grabs on and it pulls. It's almost like it's pulling a rope where it just kind of repositions, and grabs, pulls, and then pulls, and so on. So what we see is another representation of this, where we see that ATP binding, the ADP being released, and everything like that. So the ATP binding causes the release of actin from myosin. The hydrolysis causes the cocking, followed by weak binding. The ADP or the PI release causes strong binding, followed by what's known as a power stroke, where it's that movement. The ADP release causes ADP movement causes the ADP release, sorry. And then it just repeats over and over again. So those are the major components that I want you to be familiar with. Now, there are other players involved in this complicated process of muscle contraction and relaxation. For instance, the troponin complex and tropomyosin are two proteins that are also involved. And troponin 1 is represented right here. Troponin C is represented right here. And these kind of are along the lines of a actin filament. Now, what they're going to participate in is what are some of the things that cause this muscle contraction to take place. Now, contraction can be triggered by calcium influx. And if you're playing along at home, you're probably like, oh, that's Ca2 plus. And you're absolutely right. This is one of the values of being a biochemist, that you can look at an ion and say, well, it's positively charged. But it's important to also include the magnitude of that positive charge. So calcium influx into muscle cells can cause contraction. Tropomyosin binds lengthwise along actin filaments in striated muscle. This is associated with a complex of 3 troponins: troponin I, troponin C, and troponin T. In the absence of calcium, the tropomyosin troponin complex blocks the binding of myosin to actin. So this is going to prevent myosin from binding to actin, in which case the binding of calcium to TNC, troponin C, shifts the complex, relieving this inhibition and allowing contraction to occur. So basically, this troponin tropomyosin complex is going to prevent contraction of muscles. But as soon as calcium binds to it, then that contraction and relaxation can begin again. Now, when it comes to smooth muscles, contractions are regulated primarily by phosphorylation of one of the myosin light chains, called the regulatory light chain. So when we looked at the light chains previously, or the heavy and light chains, here we go, you might have seen here that we had an essential light chain and a regulatory light chain. This regulatory light chain is, as you would imagine, regulatory, and it's going to get phosphorylated, in which case, when that phosphorylation is present, then the protein can be functional. The enzyme that catalyzes this phosphorylation is called myosin light chain kinase, is itself regulated by association with calcium 2+ binding protein calmodulin. Now, increases in cytosolic Ca2+ promote the binding of calmodulin to the kinase, resulting in phosphorylation of the myosin regulatory light chain. So in essence, this is just saying that the regulation of, or the movement of myosin and actin is not simply always happening, but there are things that are going to regulate this process. Well, that wraps up day 2 of protein function. I hope that you got something from this. I think that one of the very important parts about this is the different players involved in muscle contraction and relaxation, those different discs, and the overlap of our proteins, actin and myosin, which region is just actin, which region is actin, and myosin, which region is just myosin. All right, well, I hope this is helpful, and I hope you have a good